Bismillahirrahmanirrahim, and welcome back to this next video. And this is the third video in the series of videos on the Alkaptonuria. In my last video, I've told you that because of the mutation in the HGD gene, it is going to produce a truncated kind of the protein which is totally non-functional, or it is going to produce a protein that has reduced activity. And I've told you that there are different kind of the mutations that are happening in the HGD gene, and we discussed the missense mutation as one of the example. Along with examples of the missense mutation, we discussed the nonsense mutation with examples. Another important kind of the mutations that happens in the HGD gene with reference to the Alkaptonuria, that is known as the frameshift mutation, and that is going to be the focus of this particular video. Now these frameshift mutation sometimes are also known as the indel mutations. Why we call them is the indel mutations, that will be clear in a while when we discuss this particular example. If you look at this one, this particular sequence is the original DNA. So here you are going to have the CTA, which actually codes for a codon that give you the leucine. Then you have got the AGG. This is going to give you a codon that will code for the arginine, and so on. Now, if you look at the this particular sequence over here, the CTA that remains at its place, but this particular nucleotide that has been removed, this is what this del mean. This del mean deleted. So you have deleted one base over here. Now when this base that is deleted over here, now what happens is, now the translation machinery is going to read the GGC is a new codon, and the GGC that is going to go for the glycine. So at position number 2 over here, you are having the arginine. Now at position number 2, you are going to have the glycine. Similarly, if you talk about this one, initially it was reading the CAT as the third codon, but now it is going to read the ATT as the third codon, and that is going to code for the isoleucine. So because of the deletion of the one base over here, all of the amino acid sequence that is going to change this way. All of the sequences of the amino acid that will be changed, and you are going to get a protein that most of the time is going to have a reduced activity. So this is what this del or this deletion means, and all of the frame that is shifting, and you are going to get a new amino acid sequence which is going to give you a non-functional kind of the protein. If you talk about this one, this is the original DNA sequence against this particular same sequence. I've got the CTA going to give you a codon that will code for the leucine. The AGG is going to give you a codon that is going to code for the arginine. Now what you are doing is, instead of deletion, you are inserting a new nucleotide over here. So the CTA that is going to remain at its place, it is going to code for the leucine, but now as you have insert a new nucleotide over here, the translation machinery is now going to read the GAG as a new codon, and that is going to give you the glutamic acid instead of the arginine which is present in the original sequence. Similarly, all of the amino acids down here, they are going to change again, going to give you a new amino acid sequence, a new protein with a different 3D structure, which most of the time is not going to give you the optimal activity. That is going to give you a reduced kind of the activity. So this in actually mean inserted. You have inserted a new nucleotide over here. So because of this frameshift mutation, you are going to get a different amino acid sequence, and hence you are going to get a different protein, which most of the time is a non-functional protein. Now let us discuss the frameshift mutation in the context of Alkaptonuria, and I'm going to give you a special example how the frameshift mutation that works in the Alkaptonuria. Now, when we talk about the frameshift mutation in Alkaptonuria, this is one of the example, and as I've told you in my last video, how are you going to read this particular you can say a sequence. The C I've told you, it refers to the messenger RNA sequence, for example. And if you see over here, this is the 16 to 19. Then you are having a del over here, and then you are going to have a nucleotide sequence over here. What this means is that in the coding sequence, the codon number 16 to 19, that has been deleted, and the sequence of the nucleotide that has been deleted, that is C, T, G, and T, because only 4 of the nucleotides they have been deleted. So these 4 nucleotides are C, T, G, and T. Now, what does this mean? What does this whole sequence mean? Let us discuss this. This P, as I've told you, that indicates a protein level effect. The leucine number 6, that is the normal amino acid present at position number 6. Then you have got these ALA and FS. What does this mean? Now, this ALA, that is actually referring to the alanine, and this is actually the new amino acids that appears due to the frameshift mutation because of this particular deletion. So there was no alanine in the sequence at this particular position. Now, because of the frameshift mutation, you are seeing an alanine amino acid. This Fs, that would mean the frame shift, meaning that the reading frame of the protein has been shifted because of this particular deletion. So what does this static and this 61 means? If you remember, I've told you that this static, that actually refers to the appearance of a stop codon. So this would mean this static 61, that would mean that this new reading frame leads to a premature stop codon at the 61st new amino acid position after the shift. So at position number 161, you are having an amino acid. Now, because of this deletion, this frame shift mutation, now you are going to get a premature stop codon, and that premature stop codon is going to give you a truncated kind of the protein, because the total protein, as I've told you, that is 455 amino acid. Now, if you are going to have the premature stop codon at the 61st amino acid, that would mean you are going to get a very short truncated protein, and the effect is that this truncated protein is non-functional protein, and when that is non-functional, that is going to give you the buildup of the homogenistic acid in the body that can give you the symptoms of the alkaptonuria. So this frame shift mutation, this is a specialized kind of the mutation that you see in the alkaptonuria, in the HGD gene. We will continue the discussion on the Alkaptonuria in the next video, and I'll be talking about the Punnett square.